It is now my pleasure to introduce our presenters. First, we will hear from Dr. Lei Zhang, Deputy Director in the Office of Research and Standards, or ORS, and the Office of Generic Drugs, or OGD. Then we will hear from Dr. Nilofer Tampal, Associate Director of Scientific Quality in the Office of Bioequivalence, also within the Office of Generic Drugs. Then we will hear from Dr. Myong-Jin, or MJ Kim, Director in the Division of Therapeutic Performance 2, also within the Office of Research and Standards and the Office of Generic Drugs. Please join me in welcoming our first presenter, Dr. Lei Zhang.
Thank you, Corey, for the introduction. Good afternoon. My name is Lei Zhang. I'm the Deputy Director of the Office of Research and Standards, ORS, in the Office of Generic Drugs, OGD, at the FDA. Today, along with my colleagues, Dr. Nilofer Tampal and Dr. Myong-Jin Kim, we will present to you this webinar on M13A Bioequivalence for immediate release solid oral dosage forms, implementing the final guidance. The final M13A guidance and its accompanying questions and answers Q&A document were prepared by the International Council for Harmonization of Technical Requirements for Pharmaceutics for Human Use, ICH M13 Expert Working Group, and were adopted by the ICH in July 2024. It is the first ICH guidance for bioequivalence and represents a significant milestone for generic drugs at the global level. I have the privilege to serve as the rapporteur of the M13 Expert Working Group. For M13A guidance, Dr. Tampal served as the topic lead for the FDA, and Dr. Kim served as the deputy topic lead. FDA published the final M13A guidance and its Q&A guidance 3 weeks ago on October 30th, 2024, and FDA now starts to implement this guidance. The objectives of today's webinar are, first, to provide an overview of the final guidance and the main changes from the draft guidance. Second, to provide clarifications and explain scientific thinking on main topics in M13A and the Q&A document. Finally, to take the opportunity to discuss FDA's implementation of the M13A for generic drug applications, including product-specific guidance revisions. Although M13A covers both generic and new drugs, today's focus is on generic drugs. In today's webinar, we will cover an overview of ICH M13 Guideline series, highlight M13A final guidance and the main changes from the draft guidance, provide clarifications and explanations of the scientific thinking on main topics in M13A and its Q&A, and discuss FDA's implementation of M13A for generic drug applications, including FDA's revision on product-specific guidances, PSGs, to align with M13A. Following the presentations, there will be a panel discussion with additional panel members from the FDA. We will address audience questions during the Q&A session towards the end of the webinar. Send your questions to the chat box during the presentations. Dr. Rob Lineberger, Director of ORS in OGD, will deliver the closing remarks to conclude the webinar. Generic drugs comprise a significant portion of the pharmaceutical market. In the United States, generics represent 90% of the prescription field, yet only account for 18% of the prescription drug spending. Generic drugs are often the product of a global supply chain and are produced with the intent to market in multiple jurisdictions. So common standards for global development for generics can improve the access to generic products. Bioequivalence or BE assessment is important for establishing therapeutic equivalence for generic drug products to their respective reference listed drug RLD. There may be situations also in new drug development when demonstrated BE may be critical for approval decisions. Furthermore, BE studies are used by innovator and generic drug product developers for supporting post-approval formulation and/or manufacturing process changes. The comparator is the terminology used in M13A for harmonization purposes. In the context of M13A, a comparator is the drug product accepted by regulatory agencies that an applicant can use to compare against the test product in conducting a BE study. A comparator product to support abbreviated new drug application or ANDA in the United States is the reference standard. Harmonization efforts under ICH for generic drugs started about 6 years ago following the publication of the ICH reflection paper titled Further Opportunities for Harmonization of Standards for Generic Drugs in November 2018. The M13 Expert Working Group (EWG) was formed in 2020 to develop the first ICH guideline for BE, M13, that focuses on BE for immediate release IR solid oral dosage forms. The EWG now consists of 22 ICH members and observers, including industry groups and 14 regulatory agencies. The M13 EWG is developing M13 guideline as a 3-part series: M13A, M13B, and M13C. M13A is the first guideline in the series to harmonize the scientific and technical aspect of study design and data analysis to support BE assessment for IR solid oral dosage forms. M13B, the second guideline in the series, is to cover BE for additional strength, including additional strength biowaiver. M13B was started after M13A reached step 2. M13C will cover data analysis and BE for highly variable drugs, narrow therapeutic index drugs, and complex BE study design and data analysis, for example, adaptive design. The reason to develop M13 in a 3-part series is to enable the EWG to break down topics into 3 series so that some topics may be harmonized and implemented sooner without waiting for all the topics to be harmonized. Developing M13 in series also brings efficiency in the overall process. As shown in this slide, these 3 guidelines under M13 are developed in a staggered sequential fashion following the ICH process of harmonization, which includes 5 steps. Step 1 is consensus building on the technical document. Step 2 is the endorsement of the technical document by ICH parties and adoption of the draft guideline by regulators. Step 3 is regulatory consultation and the discussion to finalize the draft guideline. Step 4 is the adoption of the final guideline, and Step 5 is the implementation of the final harmonized guideline. M13A, which is the focus of this webinar, is finalized and reaches Step 5. It is now ready for global regulatory agencies to implement. Work on M13B started when M13A reached Step 2. M13B is now at Step 1. The draft technical document for M13B is under development towards consensus by the EWG. The draft M13B guideline is targeted to be published for public comments in the first quarter of 2025. Work on M13C has not started yet. It will start in the first quarter of 2025 after M13B reaches Step 2. Now let's turn our attention to M13A. As I mentioned earlier, FDA published both M13A final guidance and its Q&A guidance in October 2024 and starts to implement this guidance. The overall content and sections of M13A in the final guidance did not change from the draft guidance. The M13A final guidance has 5 sections. Section 1 is Introduction that discusses objective, background, and scope of the guidance. Section 2 covers general principles in establishing BE with 2 main subsessions to discuss study design for PK endpoint BE studies and data analysis for non-replicate study design. The replicate study design and other more complicated data analysis will be covered in M13C. Section 3 of M13A final guidance covers specific topics with 4 subsessions. They are endogenous compounds, other immediate release dosage forms, fixed dose combination, and pH dependency. Other IR dosage forms include orally disintegrated tablets, chewable tablets, and oral suspensions. Session 4 discusses documentation, and the last session covers glossary. During the public consultation period, the M13 EWG received nearly 800 non-replicate comments. Majority of comments, that is 77% of comments received, are related to Session 2, which is General Principles in Establishing BE. It is not surprising because this is the main content of the guidance. In addition, another 10% comments are related to Session 3, specific topics. Main comment themes include clarification on high risk and when Fed-B study is needed. M13A introduced a new risk-based approach to determine whether we need 1BE or 2B studies in the fasting or fed condition or both conditions. We also received comments on additional guidance for dose proportionality assessment. This is related to the selection of dose or dose strength for the BE studies and will be expanded in M13B in terms of additional strength biowaiver. Comments also received on study population, various aspects of data analysis, group effect, and alignment of study design with product labeling. The EWG reviewed all the comments and the modifications have been made based on comments received. In addition, the EWG identified areas that need additional clarification or explanation. When the EWG finalized the guidance, they also developed a Q&A document to have additional room to provide rationale and further clarification to help the implementation. The Q&A document was published the same time as the final guidance. There were no major changes from the draft. However, modifications were made to several parts of the guidance, mainly to provide clarification or make it more practical for a regulatory agency to implement the final guidance. In particular, the high-risk products concept. What do we mean high risk? In the context of M13A, high risk refers to the higher risk of bioequivalence due to differential food effects caused by drug product formulations or manufacturing process. Data analysis part also has more clarification provided, as well as data and criteria to support PK dose proportionality assessment. For early exposure, in addition to partial AUC, Tmax may be used as the additional PK parameter. For the pH dependency section, additional clarification is provided in terms of when a BE study with an acid reducing agent or pH modifier agent is needed and what justification may be provided to not conduct such study. As I mentioned earlier, a Q&A document was published to provide rationale and further clarification to help implementation. A total of 19 Q&As were provided, and they are related to sections 2, 3, and 4. In addition, as part of the ICH process, a Step 4 presentation was developed and published on the ICH website. It can serve as additional resources and the basis for training to facilitate implementation. What's the impact of M13A? Before M13A, prior to harmonization, different regulatory agencies have different recommendations for BE study design and the criteria to support generic drug approval. For IR oral products, FDA generally recommended both fasting and Fed B studies, while several other regulatory agencies, including European Medicines Agency, EMA, generally recommended a B study under fasting conditions only. Differences in general BE guidances also had led to different product-specific guidance recommendations. For example, prior to M13A, 87% of FDA's PSGs for IR products recommended both fasting and Fed B studies, while only 16% of EMA's guidances recommended both fasting and Fed B studies. Unharmonized B study design and standards could result in drug developers having to generate multiple sets of data, leading to increased drug development time, increased drug development costs, and the lack of approval in some markets. Harmonized, M13A introduced a risk-based approach to determine B study conditions with regard to meals, depending on whether the product is high risk or non-high risk, and the dosing instruction of the comparator product. High-risk products refer to their higher risk of bioinequivalence due to food effect. For high-risk products, differences in formulation and or manufacturing process may not be detected with a single B study. Therefore, results from a fasting B study may not be extrapolated to predict Fed B study outcome or vice versa. For high-risk products, M13A recommends that B studies should be conducted under both fasting and Fed conditions. Furthermore, question and answer documents provide further rationale. On the other hand, for non-high-risk products, a single B study is sufficient to ensure there's no clinically significant difference in BE under different meal conditions. BE testing under only one condition, which is most sensitive, generally fasting condition, is sufficient. This risk-based approach represents the biggest change to the FDA's practice prior to M13A. It will significantly impact the ANDA submissions to the FDA following M13A implementation. Table on the left represents the FDA's recommendations prior to M13A, where both fasting and Fed B studies are recommended for almost all oral IR products, except for where labeling recommends the product to be taken on an empty stomach or due to safety concerns under either fasting or Fed conditions. Table on the right represents the current recommendations in M13A, where fasting and Fed B studies will only be recommended for high-risk products, while for non-high-risk products, only one B study, either under fasting or Fed condition, is recommended. The implementation of M13A can lead to significant savings for drug development. The estimated saving is about $50 million per year based on the numbers of ANDAs FDA received. For example, about 200 ANDA approvals in FY 2024 are for oral IR products that could have been impacted by these BE recommendation changes after M13A implementation. Anticipating the significant impact of M13A once implemented on FDA's previous practice, FDA took proactive measures to prepare for the implementation. For example, FDA organized training for both external and internal stakeholders shortly after the draft M13A guidance was published by holding an SBIA webinar on the M13A draft guidance in May 2023 and additional FDA internal training and awareness. FDA also strategized the process to delineate non-high-risk drugs from high-risk drugs for PSG revision. Because of the large number of PSGs that would be impacted by M13A, FDA prioritized the effort to focus on the revision on removing 1B study to provide immediate benefit. FDA also initiated revisions to the draft general BE guidance with PK endpoint to align with M13A, as well as continue the research projects on specific topics to build knowledge and further support implementation. Following the final M13A guidance adoption by the ICH, as part of the implementation, FDA published the final M13A guidance on the FDA website in October 2024. In addition, FDA revised more than 800 IR product PSGs to align with M13A. These revised PSGs represent FDA's current thinking on developing ANDAs for these ILD products. A total of 814 revised PSGs were posted in October 2024, and additional approximately 15 PSGs will be revised to align with M13A will be published in November 2024 or February 2025 with regular quarterly PS batch posting. You should check the FDA PSG forecast website for details. Also, as I mentioned earlier, the primary focus of this M13A specific PSG revision batch is to conduct risk evaluation for high-risk and non-high-risk IR drug products following the risk-based approach. The goal is to bring the most significant change to these PSGs, which is to revise from 2B studies to recommend only 1B study, either in the fasting or fed conditions. This will reduce the need for additional in vivo B studies compared to what FDA recommended prior to M13A, and it will support the streamlined global drug development. How did the FDA identify these 800 plus PSGs for revision? Currently, there are a total of more than 1,150 existing PSGs published by the FDA for IR oral drug products. Approximately 160 PSGs were excluded from this FDA's initial efforts for PSG revision if they are narrow therapeutic index drugs, locally acting drug products, products with PSGs recommending B studies in patients, or products with PSG recommending only 1B studies. Of the remaining PSGs, they underwent risk assessment. Approximately 150 of them are considered high risk, and their PSGs will continue to recommend 2B studies and will not be revised. More than 800 PSG revised under these initial efforts to align with M13A represent 72% of FDA's existing PSGs for oral products and approximately 37% of the total number of FDA published PSGs. Additional details regarding FDA's implementation efforts related to PSG revision to align with M13A and additional PSG revision efforts will be provided later in this webinar by Dr. Temple and Dr. Kim. Now I will turn over to Dr. Temple to discuss FDA's M13A implementation for generic drug applications, including FDA's PSG revisions to align with M13A. Thank you for your attention.
Thank you, Lei. We heard about the most significant change for the FDA, which is that the BE testing under both fasting and fed conditions is now limited to the high-risk category products. The rest of this webinar will focus on the FDA's efforts to align the PSGs with the recommendations in M13A. Around mid-2024, FDA put these efforts in a high gear since over 1,100 PSGs for the IR products had to be revisited. My talk will touch on the framework that we utilize to assess the risk categories, the high risk versus the non-high risk for the PSG revisions. And then for few select topics, I'll provide the rationale for the recommendations in 13A and how FDA applied those concepts for revising the PSGs. Keeping the priority in mind, which was to reduce the burden for generic developers, FDA's initial efforts focused on PSGs that recommended 2 BE studies in healthy subjects. For non-high-risk products, we updated the PSGs to recommend either a fasted or a fed study. For high risk, the PSG continued to recommend 2 BE studies, which is in line with the recommendations of 13A. Now, to evaluate the risk categories, as the first step, we designed a risk framework. This helped to guide our assessment and also ensure consistency as we delineated the high-risk and the non-high-risk products. In building this framework, we considered several factors. First is the biopharmaceutic properties of the drug substance, the BCS class, the solubility subclass, the permeability, or the fraction absorbed. Then we also considered the characteristics of the drug product, such as the complexity of the drug formulation design, the manufacturing technology and processes, and also the excipients in the formulation. Now, many IRs, we found they use simple, non-complex formulations. The high-risk designs that we identified included lipid-filled capsules, solid dispersions, and emulsifiers. We also identified pseudo-MR formulations with release-controlling polymers. With respect to the excipients, differences between formulations that would likely impact the disintegration or the dissolution, solubility, etc. were considered. For example, a formulation may have a pH modifier at a much higher level to impact the GI environment, and in such a case, a single BE study would not be considered sufficient to manage the risk. Other was the surfactants. The presence of surfactants don't necessarily make a formulation high risk. And in some instances, we may find that actually a fasting study may be more sensitive to evaluate the effectiveness of the solubilization. For example, if a product has a low solubility at all pH and has a small food effect, even with the presence of surfactant, or the size of the food effect is not clinically significant, such a formulation would not be considered high risk. Next, we factored in the dosing instructions, and this is regard to the food intake, along with the size of the food effect, and then the clinical significance of the food effect. Using this framework, we followed a 3-tier system to evaluate the risk categories. For Tier 1, if the drug product was a BCS 1 or 3, did not include critical excipients or complex formulations and manufacturing, this was identified as a non-high-risk product. whereas a drug which had complex formulations manufacturing technology, it was a high-risk product. Now, if the drug was a BCS2 or 4, or even if it was BCS1 or 3 but included critical excipients, or the information on the BCS class was either not available, it was inconclusive or conflicting, then the determination would be inconclusive, and then we would move to Tier 2. Tier 2 mainly included assessments for BCS2 and 4 drugs. Here we considered the solubility and permeability properties of the drugs. For example, for low solubility drug, was the solubility at the border of the high and low, whether it was pH dependent or independent? Was the drug product deliberately designed to modify the food effect? We considered the expected dissolution based on the solubility of the drug substance. Did that match the observed dissolution? And if not, what aspects of the formulation design were causing this difference? Similarly, we looked at the food effect, whether the expected food effect based on the BCS solubility subclass matched the observed food effect. And did this observed food effect lead to change in the dosing instructions? Taking the totality of the information that was collected, we could categorize some products as high risk, non-high risk, and then there was still, which we could not tell for sure, these were categorized as probably high risk or probably low, non-high risk. And then for this probable category, we further considered the information in the brand application, whether changes to the formulation or manufacturing of the brand drug during the development showed different food effects. If different dosage forms of the same API and the same route of administration were available, we looked to see if they showed any different food effects. And did the food effect lead to any dose adjustment? We also looked at NDAs to understand if the direction of the food effect matched that of the RLD's food effect for the different formulations. And if a formulation-related food effect was apparent, then this product would be considered a high risk. Using this framework and the three-tier system, FDA revised and posted over 800 PSGs. It's important to understand here that the PSGs represent FDA's evaluation of the risk categories for the RLD. And if a generic now uses or adds some formulation risk categories which are not found in the RLD, for example, uses a solid dispersion formulation, then FDA may ask for more information. And this may include biorelevant dissolution, PBPK modeling, or even a FED-BE study. So now, having described the risk framework and how we assess the risk categories, next I'll pivot to providing the rationales for the specific recommendations in 13A with regards to the FESTED-FED studies and how they guided the selection of the study design for the PSG revisions. Starting with the high-risk products. For these products, 13A recommends FESTED and FED study irrespective of the product labeling with regards to the food, provided safety permits. Testing under both conditions allows the product performance across the range of the GI conditions. And that's necessary for high-risk products because these are primarily low solubility products and they need to be formulated to enhance their solubility via different complex formulations or manufacturing technology. So then there is an increased likelihood that the in vivo solubility, dissolution, and absorption of these products would be sensitive to the varying GI conditions. And even if a formulation designs are similar, for example, both formulations are solid dispersions, differences in the processes or the excipients used to produce that solid dispersion could result in a different interaction with the GI conditions. And so you can understand that just a fasting or a fed alone would not be sensitive to assess the impact of these potential differences on product performance. With regards to the meal, 13A recommends a high-fat, high-calorie meal for fed BE studies, which is the same as the FDA. And the reason for this meal is that it allows to test under the conditions of extreme perturbation of the GI tract. Now, in the next few slides, we'll look at the role of the product labeling for determining the study type and the relation to the PSG revisions. If a high-risk product is labeled to be taken on empty stomach, per FDA's practice, only one study was recommended, and the currently posted PhDs also include only one fasting BE study. FDA at this time has no plans to revise the PSGs to add a fed study, unless there is a clinical concern or the labeling gets changed. We identified very small number of currently posted PSGs for the high risk, in fact less than 5 that were labeled to be taken on an empty stomach. However, going forward, FDA will consider the M13A recommendations when developing the new PSGs. For products labeled to be taken with food, either without regard to food or the labeling is silent, the existing PSGs recommend fasting and fed studies, and so no PSG revision is necessary. With regard to the meal type, again, in general, our PSGs do not specify the FDA standardized meal, the high-fat, high-calorie, for the fed studies. It's indicated in our general PK guidance, and that's the general expectation of the meal type to be used for fed studies. However, for BE in patients, FDA intends to continue to accept different fat and calorie content meals. Now moving to the non-high-risk products. For these products, 13A recommends a single study, either a fasted or a fed, and the meal can be either high-fat, high-calorie or low-fat, low-calorie for the fed BE study. The reason being that PK performance of these drugs is not expected to be impacted under the varying GI conditions. Now, if a non-high-risk product is labeled to be taken only under fasting, empty stomach, BE is recommended under fasting, which is the same as FDA's practice. The previously posted PSGs recommend a single fasting study, which aligns with 13A, and therefore no PSG revisions are necessary. If the non-high-risk product is labeled to be taken without regard to food, in general, 13A supports BE testing under fasting condition. As I said earlier, PK performance of these drugs is not expected to be impacted by varying GI conditions, meaning the state of the administration, whether it's fasting or fed, is not expected to influence the PK comparability of the products. And between fasting and fed, fasting is preferred over the fed because it's considered to be more sensitive and greater power to discriminate between PK profiles. Our previously posted PSGs recommend 2 studies, fasting and fed, and these were therefore revised to remove the fed study. Next, we look at scenarios where the product is labeled to be taken with food. First, when food is recommended for PK reasons. In this case, a single fat study is sufficient either under high-fat, high-calorie or low-fat, low-calorie meal. The flexibility of the meal type is allowed because, again, the variability across the GI is not expected to significantly influence the performance of these products. For non-high-risk products, it is not really necessary to test under extreme perturbation conditions, which is provided by the high-fat, high-calorie meal. But M13 also does not preclude the use of a high-fat, high-calorie meal. Testing under less severe GI perturbations, such as using a low-fat, low-calorie meal, is also acceptable. And in fact, it may better reflect the type of the meal a patient is likely to consume. we understand that there is no single meal type that can represent the diverse range of the meals a patient may consume. Now, with respect to the meal type for non-high-risk products, FDA's recommendations for high-fat, high-calorie meal does not reflect the same flexibility which M13A provides. So FDA will consider the recommendations of alternate meal types in M13A for new PSGs and also in our ENDA assessments with adequate justification. Now, the previously posted PSGs with 2 BE studies were revised to remove a fasting BE study. The second scenario is where the non-high-risk product is labeled to be taken with food for tolerability reasons. M13A recommends, again, for this purpose, either a fasted or fed study. Of the two, fasting would be the preferred option since dosing with food here, in this case, is recommended primarily for a non-PK reason. And tolerability from chronic use may not be a concern for a single use. For example, if the GI irritation is minor from a single use. Now, FDA applied some judgment to the PSGs. We found that for approximately 10 PSGs, the products were labeled to be taken with food, but we already recommended a fasting BE study because fasting may be more sensitive for various cases. If the previously posted PSGs included 2 BE studies, then they were revised to remove the fed BE study. Now, another major recommendation in 13A is related to drugs with pH-dependent solubility. If these products are expected to be taken with an acid-reducing agent like a PPI, then 13A recommends an additional study with concomitant administration of the PPI. If the comparator was explicitly designed to overcome the effect of the altered gastric pH using a pH modifier in the formulations. Now, besides the pH modifying excipients, even differences in salt or polymorphic forms or differences in the manufacturing process between two formulations may affect the pH-dependent solubility differently. So the impact of sustained increase in gastric pH on the solubility and the dissolution, and therefore the drug availability, may be underestimated from a fasted and fed study alone. And an altered bioavailability in this case can be of clinical significance, specifically for drugs which are either weak base drugs or the weak acid drugs. With regards to the PSGs for these drug products, FDA's initial efforts do not cover these PSGs. In recent years, FDA has recommended an additional study with PPI in the PSGs as needed. For example, we have the palbociclib tablets, the PSGs for the palbociclib tablets. For new PSGs, FDA will consider the recommendations in M13A going forward. If applicants want to pursue alternate approaches, both FDA and M13A provide this flexibility. And some examples, including the comparative dissolution and disintegration in biorelevant media and modeling such as the PBPK, are identified in M13A. If proposing an alternate approach, appropriate scientific justification should be provided. For example, if a product is formulated to be qualitatively and quantitatively similar to the comparator and uses the same manufacturing technology as the comparator, applicants may be able to propose a waiver for 1BE study for a high-risk product. In all cases, the prospective applicants need to provide the rationale for the selection of the study design with regard to the study type and the meal. This expectation is specified in M13A. Applicants are expected to have a thorough understanding of their product under development, and they should consider if their product has the potential for formulation-dependent food effect, and then accordingly select the study design to demonstrate bioequivalence of their product. As I mentioned earlier, The study design in the PSGs are based on the understanding and the information of the reference product, and they may not always apply to your product. In the next 2 to 3 slides, I will address some commonly asked questions. We have received several variations of these questions, but the theme seems similar. The first set of questions are related to, we conducted both fasted and fed studies, but now the PSG recommends only one study. Should we submit the not recommended study? And what if that study has failed? Should we still submit? The short answer to this is a yes. FDA's regulations require the submission of data from all BE studies which the applicant conducts on the same drug product formulation as that submitted for the approval. And this includes the pilot studies, the pivotal failed studies, in addition to the pivotal passing studies. Also in M13A, in the documentation section, it recommends to submit all relevant BE data conducted regardless of the study outcome. And to understand what we, the FDA, considers as the same drug product formulation, I'll refer you to the guidance noted at the bottom of the slide. The theme of the next question is what type of study reports need to be submitted. For example, in this case, if the FED study is removed from the PSG, should a complete report for the FED BE study be provided, or would only a summary of the FED BE be sufficient? So again, a complete report is needed to be submitted for BE studies upon which the applicant relies for approval. So for the pivotal BE studies, a complete report is expected. For each additional study conducted on the same drug product formulation, such as the FED study in this case, either a complete or a summary report should be submitted. That would mean the BE summary tables in Module 2.7. And if, while reviewing that summary report, if FDA believes that there may be BE issues or concerns with the drug product, then FDA may request for a complete report. And again, the summary report, for the summary report, I'll refer you to the guidance here noted at the bottom of the slide. And the next one is, if the PSG recommends only a fasting BE study, and our application was already submitted with both fasted and fed studies, how will FDA treat the additional fed study for the purpose of drug approval? So, based on FDA's standard practice, we review the data from all studies in the application, especially those which are conducted on the same drug product formulation. In this case, the FED study may receive an abbreviated review, and most likely FDA will not ask applicants to repeat the study that is not recommended in the PSG, unless the evaluation of the data raises concerns about the performance of the drug product. So I hope these questions help to some extent. And now I'll turn it over to Dr. Kim to talk about the additional M13A topics and the PSG revisions. Thank you for your attention.
Thank you, Nilofer. In Nilofer's talk, she presented the points for consideration for delineating the risk categories for IR drug products as high risk and non-high risk based on the M13A guidance. From here on, my presentation will cover additional M13A related topics, as well as the other non-M13A related areas that are reflected in the October posting of PSG revisions. In anticipation of publishing the M13A final guidance in 2024, the agency has dedicated significant efforts to revise the previously published PSGs of IR drug products to align with the M13A recommendations. A significant change was the implementation of the M13A guidance is the removal of recommendations for BE studies conducted under either fasting or fat conditions. While the FDA generally publishes a quarterly list of PSGs that we plan to revise in the coming year, given the considerable impact this revision has on generic drug developers, we posted a list of PSGs along with the planned revision category and description of the potential changes in September 2024. In October 2024, a total of 814 revised PSGs were published, changing the recommendation from 2 BE studies to 1 study. In addition, the FDA PSG forecast website was updated to reflect the modifications from the planned revisions in the PSGs, including any changes to previous September forecast posting. For example, if the planned revision was to remove a fasting BE study, but the recommendation for a fat BE study was removed instead, or if no changes in the recommending both fasting and fat BE studies were made, this information was included in the modification section. Additional 10 revised PSGs are planned to be published between November 2024 and February 2025 with the regular PSG batch posting, and these PSGs are forecasted in the FDA webpage. As I briefly mentioned at the beginning, there are other PSG revisions not directly related to the M13A guidance, which are included in the September 2024 forecast list under the planned revision category along with a description. This includes the addition of a BCS-based biowaiver option categorized as a minor revision. Additionally, revisions to language and formatting as needed were made, and they are categorized as editorial revision. In the October 2024 posting, a BCS Class 1 or 3 based biowaiver option was added to certain PSGs that were not included in the September forecast description. The upcoming PSGs for generic drug product development webpage has the following table format of the planned revised PSGs to align with the M13A guidance. Similar to the FDA's regular PSG batch forecast, which is updated quarterly, the planned revised PSG forecast associated with the M13A implementation includes the following list: active ingredient, route of administration, dosage form, RLD or RS application number, planned revision category with description, product complexity, and planned publication date. However, the posting table includes an additional column that explains the modifications made to the originally planned revisions in the PSGs. As examples, when reviewing these two drugs, the last column indicates a BCS Class 1 based biowaiver option was added, and the recommendation was to remove a fat BE study instead of a fasting study. For revised PSGs, there are 4 different planned revision categories, and they are critical, major, minor, and editorial revisions. It should be noted that all M13A related PSG revisions in October 2024 were categorized as minor revisions. For your information, the minor revision is defined below. In addition to the fasting and fat BE study related recommendation, there are other M13A related revisions reflected in the PSGs published in October 2024. One example is the revision of chewable tablet and ODT PSGs regarding the administration of a drug with or without water. For ODTs, the M13A recommends that ODT should be administered in BE studies according to the RLD labeling with regard to intake of water. If the RLD labeling states that the ODT can be taken with or without water, the test and the RLD should be administered in the BE study without water. as this is considered to be more discriminating scenario. One example of previously posted ODT PSG states that water may be taken after 5 minutes and includes a reference to using liquid. In this case, the PSG was revised to specify that ODT should be administered without water and the word liquid was removed. For chewable tablet PSGs, if the previously posted PSGs were unclear about administration, specifically whether to chew or take with water, they were revised to state that chewable tablets should be chewed and swallowed without water. In terms of study population, the 2021 General BE guidance defines healthy subjects and general population as separate terms, emphasizing that they are not interchangeable. To align with the M13A guidance, we removed the terms general population, normal, or healthy and general population, and updated the PSGs to specify healthy subjects. Other revisions include risk to females of reproductive potential. It recommends that the risk to females of reproductive potential be considered and that pregnant or lactating females should not be included. Male contraception is recommended if a drug poses embryo-fetal toxicity risk and may affect a female partner of reproductive potential. The 2021 General BE guidance recommends that if a drug product is intended for use in single sex, then the applicant should only include subjects of that sex. As a result, we identified a list of PSGs for drug products intended for use in a single-sex population and revised the study subject recommendations accordingly. For example, for PSGs of drug products intended for use in females only, the study population was revised from healthy males and non-pregnant females general population to healthy non-pregnant, non-lactating females. Some of the examples include a combination oral contraceptive for prevention of pregnancy and a progestin indicated for the treatment of secondary amenorrhea, endometriosis, and abnormal uterine bleeding due to hormonal imbalance. This table summarizes the recommendations for study subject populations in BE studies with PK endpoints, as outlined in the M13A guidance, previously posted PSGs, and the updated PSGs released in October 2024. As previously mentioned, we removed the terms general population, normal, or healthy and general population, and updated the PSGs to specify healthy subjects. If the drugs are intended for use in both sexes, we have maintained our previous recommendation to include both sexes in the study population. For drug products intended for use in a single-sex population, we recommend including subjects of that specific sex. This revision is reflected in several of the PSGs published in October. A noteworthy update is the inclusion of a dedicated section in the M13A guidance addressing risk to females of reproductive potential. It recommends that the risks to females of reproductive potential be considered and that male contraception be advised if drug poses embryo-fetal toxicity risk and may affect a female partner of reproductive potential. In contrast, earlier PSGs, particularly those published before 2015, often did not address these considerations. However, more recent PSGs now reflect these safety concerns, and the older PSGs have been updated accordingly. Depending on the level of safety concern, we now recommend including healthy females not of reproductive potential, healthy males not of reproductive potential, or advising male contraception. While revising the 814 PSGs for the October posting, We also took the opportunity to address other areas where applicable. These revisions include adding a BCS-based biowaiver option, recommending the use of reference-scaled BE approach for highly variable drugs, and revising the inclusion or removal of risk evaluation and mitigation strategies. As mentioned in the previous slide, other revisions incorporated in the most recent publications of the PSGs include the addition of the BCS Class 1 or 3 based biowaiver option. For example, we added a biowaiver option to the BCS Class 1 drug PSGs that a waiver request of in vivo testing may be considered, provided that the appropriate documentation regarding high solubility, high permeability, and rapid dissolution as detailed in the M9 guidance is submitted in the ANDA. Another example involves drugs classified as highly variable. For this, we have added the use of a reference-scaled average B approach. While the principles of these approaches and recommendations are already included in the FDA's general BE and M9 guidances, prospective ANDA applicants may find it helpful for this information to be clearly stated in the PSGs. In addition to PSG revisions related to BCS Class 1 or 3 based biowaivers and the use of a reference-scaled average BE approach for highly variable IR drug products, we have made other PSG revisions that address safety-related topics. We assessed the FDA's current REMS public dashboard and updated our previously posted PSGs to reflect any changes based on the most current information. We added REMS language to the opioid drug product PSGs, with one example being the oxycodone hydrochloride tablet PSG. We added a statement that this drug product is under a REMS with ITASU, and therefore all pertinent elements of REMS ITASU should be incorporated into the protocol and informed consent. 4 PSGs were updated by removing the REMS language because either REMS or ITASU is no longer required or recommended. Another safety-related concern is embryo phytotoxicity or fertility impacts. To mitigate these risks, we recommended selecting healthy males who are not of reproductive potential or healthy females who are not of reproductive potential as the study population. In our efforts to implement the M13A guidance by aligning the PSGs with its recommendations, we are conducting ongoing assessments of the several topics, including pH dependency, dose or strength to be studied, which analyze to measure, and the risk assessment of certain products, such as liquid-filled lipid-based capsules and solid dispersions. We recognize the importance of safety considerations when recommending study subjects in the PSGs. We are continuing to assess whether patient-based or healthy subject-based BE studies should be recommended, taking into account all available information to make appropriate recommendations. We are aware that there are differing recommendations regarding study population, type of studies, and mere conditions. Additionally, we acknowledge that specific subsets of healthy subjects, such as sex, age, menopausal status, or reproductive potential, may need to be reconsidered. While our efforts to implement the M13A guidance are reflected in the most recently published PSG revisions from October, some previously published PSGs, such as those for the high-risk drug products with one BE study recommendation, patient-based BE study recommendations, and drugs exhibiting less than dose proportional increases due to solubility or when proportionality is unknown, remain unchanged and were not revised. As stated at the beginning of each PSG document, our currently published PSGs reflect the FDA's current thinking, and the prospective ANDA applicants should continue to rely on the PSG recommendations. If any revisions are considered, the agency will provide a forecast of future revisions, including the category and a brief description where applicable. As mentioned in the previous slide, the FDA's PSGs reflect its current thinking on a topic, and therefore prospective ANDA applicants can utilize the PSG recommendations. This is reflected as the opening statement in each PSG page. If prospective applicants propose alternative approaches to the M13A guidance or PSG recommendations, they may be acceptable if appropriate scientific justification is provided. For seeking the FDA's guidance, applicants may submit control correspondence or request meetings through the appropriate pre-ANDA or ANDA meetings, such as PSG teleconference and PSG meetings as applicable. For PSG teleconferences, either pre-submission or post-submission options are available as appropriate. Please refer to the relevant guidances for details on these meetings, or consult the previously offered SBI webinars on these topics. With respect to PSG-related communications, the FDA posts plans for issuing new or revised PSGs on its website, including information on upcoming new and revised PSGs in the next 12 months. The website is updated each time a new quarterly PSG batch is issued, with the updates removing published PSGs and adding those under development or revision. This advance notice should help prospective ANDA applicants with their drug development efforts. Regarding public comments on PSGs, the agency seeks feedback and considers comments submitted to the docket for these guidances. Comments can be submitted to the docket or by mail at any time. With that, here's one challenge question. What is the expected impact of the final M13A guidance on BE studies for generic drugs? A. Reduce the number of studies recommended. B. Increased emphasis on in vitro BE studies. C. More stringent criteria for BE acceptance. D. No significant changes to current BE study protocols. The answer is A. Reduced number of studies recommended. The next two slides summarize the key takeaway points. We are excited that the M13A, the first harmonized ICH guideline that focuses on bioequivalence, is now finalized and that the FDA is ready to implement for generic drug development. The significance of M13A implementation is truly noteworthy given that IR drug products account for the considerable portion of the pharmaceutical market in the US. The implementation of the M13A guidance will reduce the need for additional in vivo BE studies that arise due to divergent regulatory recommendations before harmonization. It supports the streamlined global drug development, ultimately benefiting patients by increasing access to generic drugs. As part of the agency's efforts to implement the M13A guidance, we are updating the relevant guidance documents to align the recommendations in the M13A final guidance. This includes the agency's latest efforts to update the PSGs for IR drug products, For those IR drug products classified as non-high risk, either a fasting or FAT-B study is recommended. Additionally, the agency plans to publish the updated General BE guidance in 2025. The currently published PSGs reflect the FDA's current thinking, and prospective ANDA applicants can continue to reliably use the recommendations provided. Prospective ANDA applicants may propose alternative approaches to the M13A guidance and PSG recommendations, provided that they submit appropriate scientific justification. And lastly, applicants may engage with the agency through the appropriate meetings or control correspondence. Lastly, this slide shows the relevant resources for your information. With that, this concludes our SBI webinar presentation on the M13A bioequivalence for immediate release solid oral dosage forms implementing the final guidance. Thank you very much for your attention.
Thank you for all our speakers today. I'd like to go ahead and welcome our speakers back to our panel discussion, and they will be joined by Robert Leinberger, our Director for Office of Research and Standards, Partha Roy, Director of Office of Bioequivalence, Dave Coppersmith, Regulatory Counsel and Division of Policy Development in the Office of Generic Drug Policy, and Lei Zhang in the Immediate Release Drug Products Team Leader in ORS as well. So welcome, everyone. Our goal today was to explore the practical aspects of implementing M13A and its integration with existing FDA guidances. And we will begin the panel discussion with our first questions regarding M13A and its implications. Following that, we will open the floor to audience questions. So please document what it is that you would like to ask us and what you would like us to address. So we can move forward to our first question. So our first topic really focuses on FDA's approach to implementing M13A alongside existing guidances and its alignment with the upcoming M13B and M13C guidelines. Lei and Dave, can you comment more on what is FDA's approach for implementing M13A with other existing FDA guidances, and how does this align with the future M13B and M13C guidelines?
Sarah, thank you for the question. So the main currently published FDA general guidance that is impacted by M13A is the bioequivalent studies with pharmacokinetic endpoints for drugs submitted under an ANDA guidance, the current version of which was published back in August of 2021. And that guidance is also referred to as the ANDA bioequivalence guidance, and that was referenced in the presentation as well. Most of the topics that are discussed in M13A for immediate release solid oral dosage forms are also discussed in the ANDA bioequivalence guidance. However, the ANDA bioequivalence guidance does have a broader scope than M13A and discusses some topics that are not discussed in M13A. So for example, the ANDA bioequivalence guidance discusses modified release drug products, narrow therapeutic index drugs, and highly variable drugs. But now that M13A has been finalized and adopted by FDA, it represents the agency's current thinking on that topic. And as noted, FDA is in the process of revising the ANDA bioequivalence guidance to align the recommendations for immediate release solid oral dosage forms with the recommendations in M13A, and to ensure consistency in the recommendations between the two guidances. FDA is also making other revisions to sections in the guidance in response to comments that were submitted to the docket when the draft guidance was published back in 2021, and to make other updates deemed necessary by the agency. In the future, as M13B and then M13C are finalized and adopted by FDA, FDA intends to make additional revisions to the ANDA bioequivalence guidance to align the recommendations with those two guidances. Thank you.
Thank you so much, Dave. Lei, can you comment, please?
Thanks, Sarah. Yes. So as I present in my presentation, M13A is developed in series for a reason, and also perceive the benefit allow us to in some topics sooner rather than waiting for all the topics to be harmonized, which will take a longer time. For example, now M13A is final, which allows different regulatory agency to decide, and it's covered under M13A. I understand the different have been planned for implementation. As heard, it's to implement M13A right after it is finalized without waiting for M13B and M13C to be finalized. This brings immediate benefit to the industry by only need to conduct 1B study for a majority of the IR product that are considered to be non-high risk. In the interim, like the topics covered by M13B, M13C, or even modified release products that are covered by FDA will not be changed. So you should look at FDA's genetic program until it's finalized or harmonized. And we are in the middle of revised genetic guidance to at least update
Lei? We're having some technical. Will you mute your phone, your microphone, please?
Say that again.
Can you mute your microphone?
I'm talking right now. No, I should mute my mic.
Mute and unmute again. Let's see if the connection works this time. There was a connection issue.
Which part? Or I just don't know when the issue occurred.
You sound better now. You may just want to summarize it because we really don't know where you cut off.
Okay, I just want to say, anyway, in the interim for topics covered by M13B, M13C, or relate to modified release products, you should follow FDA's general NDB guidance. And we will revise those part of the general guidance until those topics are harmonized under ICH. And we also, FDA do have a separate biostats B guidance that cover additional topics that may not be under M13 scope. We recognize there may be some confusion in the interim. So if industry has questions, they should always reach out to FDA, and the FDA do offer flexibility. If you found you have confusion or you take an approach deviate from our general guidance or PSG, we hope our PSG already being up to date, reflect our current thinking. But if you do use alternative approach, can provide the justification on your approach in your submission. Yeah, that's all.
Thank you so much, and apologies everyone for the technical difficulties. Moving on and building on that to our next question, Nellifer, let's talk about the implications for ANDA submissions and how does the FDA intend to apply M13A impacted PSG revisions to pending or soon to be submitted ANDA applications?
Yeah, Sarah, thank you for the question. Yeah, so for applications that are under review or they are soon to be submitted applications, if you have, where 2 studies would have been conducted, but now the PSG is recommending only 1 study, in that case, we will be following our currently posted recommendations in the revised PSGs, I should say. If your product is also a low risk, like the reference product, then 1 study would be sufficient. Now, from a review perspective,
Since the study, that is the recommended study in the PSG, will be a pivotal study, it will be reviewed no different from how we would normally review our other pivotal studies. And for the not recommended study, it would be treated like an additional study. And we will perform an abbreviated review for that study, which is also our standard practice for reviewing the additional studies. And we are probably not likely to communicate any deficiencies related to the not recommended study unless we have identified certain bioequivalence concerns with the drug product or the formulation itself. Thank you, Sarah.
Thank you so much, Nelifer. And just to build on that, Partha, how will the M13A impacted PSG revisions impact bioequivalence studies under review for data integrity?
Let me just say, can you hear me? Okay, yeah, so I know data integrity has come up many times, and it affects all our work and for the last few years. So obviously one of the things we would see is that if the data integrity lies with the study that is not the required study based on the M13 revision, then obviously that data, we think, looking at the product itself, and as a, I'm guessing it's a non-high risk product, and I mean, sort of the high risk product, but a non-high risk product that you need only one study. In that scenario, the non-required study, if the data integrity is there only, then obviously, you have a less concern in that particular application. Our overarching principle is that when you redo the study or when you resubmit the study, it's really you have to address the requirement and the recommendations as it stands today at the time of resubmission. So at this time of resubmission, if you're talking of only one study, and that's a clean study which has been done in CRO, which is not impacted, then you are, in general, you are good to go, unless we have other concerns within the application, obviously. Then of course we will review it. So it's more like and there are some companies probably in that scenario. I mean, we don't see that there will be a lot of companies applications which have, like, usually people do their fasted and fed studies both in one CRO. I mean, for obvious reasons and logistics issues and things like that. But if it is a situation where the studies have been done in different CROs and one of them get impacted and the other not, we obviously will focus on the required study moving on. So that's sort of the answer.
Thank you so much, Partha. And moving on to our next question. And this is more directed to Chi and MJ. What are the key criteria used to classify a product as high risk versus non-high risk? And what specific data are used to guide PSG revisions?
Yeah, thanks, Sarah. So perhaps I can kind of start the discussion, and maybe the Chi can kind of elaborate a little more on this. So Chi is the expert in our team, immediate release team, who's been working extensively on this topic. So she's really qualified to give her perspective. But when it comes to all this 814 plus PSGs, when we were actually looking at the list started with more than 1,000 as a previous slide showed. So we looked at all those immediate release drug products, PSGs that we have published up to now. And what we did was we looked at all those available information on the reference listed drug products, including the product labeling, to see what type of information we can gather. And also we looked at the formulations and the food effect impact. So we assessed all that information for the preparation of the October posting. So with that, Chi, maybe you can give a little further elaboration on this topic. Thank you.
Yeah, can you hear me? Okay, yeah. So yes, thank you, MJ, for the overview, insightful overview. I like to delve a little bit specifically on the rationale behind for the risk assessment and the study conditions we recommend. And so as now outlined in our presentation, we use a systematic framework and a data-driven approach. As part of this framework, we carefully evaluate elements like biopharmaceutics classification system, product complexity, including formulation and manufacturing process, and also dissolution profiles and food effects and clinical considerations to inform and guide our decision. So those factors are integral during our triage process and risk evaluation and further investigation, ensuring our recommendation align with the level of the risk in the framework. I can walk through some details. We look at the complexity of the product's formulation and the BCH classification. For example, BCH class 2 and 4 drugs with low solubility or drugs with uncertain solubility profiles. And those drugs tend to show variability in between fasting and fat. Then often the product is formulated with specialized excipients or advanced manufacturing techniques to enhance the API solubility or product dissolution. In contrast, the APIs with high solubility and high permeability are typically low risk because they formulate most of the time with a standard formulation because of the high solubility and the fast dissolution. And for BCAs with low permeability and then low solubility, sometimes they may also have some simpler formulations like conventional tablets and capsules with standard excipients such as fillers and binders can be also classified as the low risk because they can show more predictable performance in vivo. And dissolution is a key factor for this evaluation. So the high-risk products may show inconsistent dissolution across dosage forms or strengths, and they may have some mechanisms like a slow release for the IR products that require additional evaluation. And the low-risk products, however, typically they have very consistent dissolution according to their biopharmaceutics classification. Therefore, they have more predictable in vivo performance between fasting and fat across all the strains. So we look at how those products behave in the presence of food, and high-risk products often show very significant or unexpected food effects from their business classification. Such as a large reduction or a large increase in terms of food effects and absorption, or there's inconsistent bioavailability between fasting and fat conditions. And low-risk products, by contrast, exhibit more predictable food effects that align with the API and characteristics. So make it easier for us to make assessments and recommend one specific study condition. So overall, the product that is simple in product design and uses standard excipients and contains a highly soluble API and exhibit consistent dissolution and predictable food effects is considered low risk. For those products, a single B study, either fasting or fat, may be sufficient. In contrast, the product with a complex design, specialized excipients, or the API with uncertain solubility and combined with inconsistent food effects, large food effects, or inconsistent dissolution profiles may be classified as high risk. Those products typically require both fasting and fat studies to fully understand and evaluate the variability between fasting and fat to make sure the therapeutic equivalence. So the framework the FDA currently are using evaluates not just the API, but also the products, the excipients, the formulation, and also consider real-world use of the products. So each tier will add a layer of the complexity, and the cumulative assessment determines whether the product is classified as high or low risk, therefore guiding our PSC recommendation and study requirements. Yeah, that's a perspective from my end. Thank you.
Thank you so much, Chi. Moving to Rob, what would, and moving to the next question, please, directed towards Rob, what would make a generic product a high risk, and how does the FDA apply this risk-based approach to determine the recommended BE studies for ANDA approval?
So as was mentioned before, our PSG recommendations are primarily based on the reference product, right? So generally, that's what determines our recommendations. And so at the time we write the PSGs, generally we try to write them soon after the RLD is available, so there won't be generic products available at the time we write the PSGs in the future as well. And so the question is, what would a generic applicant have to do if FDA has said in our product-specific guidance that the reference product is a low-risk product? Is it possible for the generic product to be a high-risk product? And the answer, I think, and this is something that has been mentioned before in the presentations. You as a generic applicant, if you do something unusual, you can make your product high risk even when the reference product is not. And so, an example of that would be using an amorphous dispersion, right? So if the RLD is a conventional formulation, and for some reason you're manufacturing your product with an amorphous dispersion and precipitation controlling polymers, that would put that in the higher risk category. But again, in most cases, we've looked across the types of products that have been submitted to us in many generic applications. Generally the generic industry is not sort of introducing novel technologies when they're not needed to meet some performance aspect of the RLDs. So in general, we usually will see that if the reference product is a conventional formulation using standard excipients in their normal way, and the generic product uses, again, a conventional formulation using standard excipients in the reasonable and expected way, then, both products would be low risk and our product-specific guidances, would apply. So again, we think that would be the vast, vast majority of the cases. It's very rare that we can look and say, well, look, here are cases in there are thousands of products that have been submitted to us, cases where the generic company did something very different. But again, there are rare situations where, there may be some business reasons for that or product quality reasons to do that. But, in general, it's introducing some sort of novel formulation or manufacturing technology that's significantly different from the brand product, not a conventional formulation at all. And that's the kind of place where, you may, in that case, get more questions from the bioequivalence reviewer. But it may not, even in that case, it may not mean that you have to do another study. It may not provide more information about what you did, maybe some justification for it. But again, I think we do believe that situation will be rare, but that's something that, we're, from the learnings that we've made as we've gone through identifying which of the RLDs are higher risk, we have a list of, factors and things to consider for our review staff as well. But again, we think it's actually very rare that a generic company would try to add in some sort of novel technology or surfactants that were not contained in the reference product without any particularly solid reason for that. We think that's just a rare occasion.
Thank you so much. And this concludes our panel discussion and we will be moving into the questions from the audience. And to really summarize, M13A offers a harmonized approach to bioequivalence studies with significant implications for PSG revisions and ANDA submissions. And understanding the FDA's risk-based framework is critical for classifying high-risk versus non-high-risk products and determining the appropriate study types. And we cannot stress this enough, collaboration and early dialogue with the FDA remains key to navigating these updates successfully. And with that, we can go ahead and move into the Q&A from the audience. Corey?
Yes, thank you, Sarah. And thank you again to our presenters and panelists for the excellent presentations and discussions. We'll now begin our question and answer panel. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We will answer as many questions as time allows. So for our first question, this one is for Lei. Can you please clarify in which situations BE is critical for approval of a new drug, given that the FDA guidance doesn't require demonstration of BE pre-approval, only for post-approval changes? Are there any other HA requiring BE?
Thank you for the question. Yes, the guidance did talk about there might be some situation, even pre-approval for new drug, the BE may be critical for decision making. This could include sometimes BE may be used for bridging the formulation during the development phase or for certain 505(b)(2) application to the FDA. However, this is a case-by-case situation, and it will be, I think it will be best for you to reach out to the appropriate review division at the FDA for your specific question, and also the other regulatory agency. As I mentioned in my presentation, this webinar mainly focused on the generic drugs, but for new drugs, you can always reach out to the appropriate review division. Thank you.
Thank you. My next question is directed towards Partha and also MJ, if you'd like to also join in with this one. The question is, regarding the recommendation of inclusion of both males and female subject in the BE studies, is there a particular portion of each sex that we need to consider, or can we just include a limited number of females in the study? If we have an all-male study, can we provide justification of an all-male study?
Yeah, so thank you very much. Let me start saying that I know this issue has caused a lot of heartaches and headaches, particularly since our ANDA-PK guidance was published. The general guidance was published, where we commented on the need to enroll both males and females in the study, and that's not really a change in policy. I mean, we have always encouraged applicants to enroll both the sexes in the study for a variety of reasons. And also, I think in that particular guidance, the first time we said, please provide the justification if you are enrolling a single sex population, or in other words, all male study. So we'll continue to have that posture. That is, you need to provide the justification if your study is all male study and the drug is actually approved for both the sexes. Let me also articulate what we really do not want is to actually set up a protocol saying that we will only enroll males for the study, unless there is, of course, a safety issue or the product is exclusively indicated for one sex. So definitely in our own normal assessment process, we closely review the protocols to make sure that it is not set up that way, and you are doing a proper due diligence to enroll both the sexes, and most of the applicants actually continue to do that. I know M13 probably did not elaborate a lot on this topic, but our general guidance is very much aligned with the M13, saying that we would like both males and females for BE studies for the products that are intended for both the sexes. Now I'll transfer over to MJ.
Yeah, thank you, Partha. And looking at the questions that we are receiving here, there are a lot of questions on this topic. So it seems like the audience here, you're very much interested in this. So some clarification, and just by reading those comments and questions in the chat box. So first of all, the 2021 general BE guidance, yes, it does mention of a similar proportion of males and females if a drug product is indicated for use in both sexes. And yes, M13A guidance, if you take a look, it just says males and females should be considered, and we don't say the word similar proportion. And then you also heard from Lei's presentation as well as mine that the general BE guidance, the updated version, will be published in 2025. And the version that's going to be published in that year, next year, will be in alignment with what the M13A recommends when it comes to both sexes. So just kind of going back to your overall questions, if a drug is intended for both sexes, yes, we would like to see both sexes be included. But once again, we don't specify what the percentage and all that. And if you have a justification as to why only one sex is included in the BE study, please include. But always be mindful that for certain drug products, if you look at the drug product labeling, it may talks about how there are PK differences in males and females. So you need to consider that aspect. And also, lastly, if a study design that you're considering a parallel study design, of course, we'd be interested especially when there are PK differences in males and females. So with that, thank you so much.
Thank you both for that excellent response. My next question is for Nilofer. If a high-risk product, RLD, that is labeled to be taken only with food, is it necessary to perform BE study in fasting or fed conditions?
Yeah, thank you for that question. So yes, as I also covered in the my presentation, if you have a high risk product, we do recommend studying under both fasted and fed conditions, and that's because of the complexity or the manufacturing technology that may be used. And it's also the reason to be used is to enhance the solubility and the absorption of that drug product. So we want to kind of test under all the different conditions of the GI physiological conditions under the fasted and fed states, so that assures us of the bioequivalence. So yes, the short answer for the high risk products, we ask for fasted and fed studies. Thank you.
Thank you for that response. My next question is directed for Dave. Prior to this PSG revision, we performed both studies under both fasting and fed conditions for one of our products. Now that the PSG has been revised, can we submit only the fasting study?
Thank you for the question. So FDA's regulations require that applicants submit all information from all bioequivalent studies conducted with the same formulation of the proposed drug product. So in the situation you described, you would need to submit data from both the fasting study as well as the fed study. And you can refer to FDA's guidance for industry, submission of summary bioequivalence data for ANDAs, which was published back in May of 2011 for more information. Thank you.
Excellent, thank you. My next question is directed towards Robert here. Since these are draft PSGs, is there a chance of rolling back to waiver of fed BE studies later on?
I mean, generally, no. We've gone through very detailed discussions through the many years of the M13 process, right, to establish the scientific foundations and the global consensus around what the most appropriate bioequivalent studies are. And then the PSG updates, again, are looking at each specific drug to say how those general recommendations in the M13 guidance and process came out of that. So again, I think, so certainly, if we learn new information about particular products or risk factors, those can change our guidance in the future. But we generally wouldn't make these changes unless we believe that they were the best current recommendation.
Excellent, thank you. My next question will be directed towards Chi here. The PSG recommends 2 studies, fasting and fed. Does that mean this is a high-risk design formulation?
Yeah, my answer is not necessarily. So the recommendation for both fasting and fast studies is not solely indicate of high-risk formulation design, but rather reflects the need for comprehensive BE evaluation of the product's behavior and the different food conditions. So while the high-risk formulations often require both studies due to their complexity, such as nanotechnologies and lipid-based or ASDs, and the other factors actually may also warrant the dual studies. For example, if there is a formulation-related food effects, whether the product's absorption bioavailability changes in the present food, fat studies may be also recommended to ensure consistent therapeutic performance compared to the innovator products. So even with the API with predictable behaviors, for example, B6 class 1 or 3, and formulation-specific factors may also influence the product's dissolution absorption. So there are some other considerations too, such as clinical relevance and pediatric use projects. Those may be recommended even for the low-risk products. Yeah, that's my answers. Thank you.
Excellent. Thank you very much for that response. My next question is for Lei. And so for this question, when an innovator has filed a petition recommending BE study with clinical endpoint and PSG is also available, which recommends a BE study with a PK endpoint, should the ANDA applicant perform a BE study with both PK and clinical endpoints?
Yeah, thank you for the question. In this regard, it depends on what the FDA's final decision on the citizen petition is. We will either remain our recommendation for the PSG, or we may modify the PSG based on the citizen petition. But until that revision is made, the PSG will be reflect our current thinking. The applicant should follow the PSG. Again, if they want to deviate from the PSG, they can provide scientific justification for FDA to consider. Thank you.
Excellent, thank you. My next question is for Nilüfer. Some of FDA's new PSGs state recommended study, one in vivo bioequivalent study with pharmacokinetic endpoints. Is there a thought pattern to specifying pharmacokinetic endpoints instead of just one in vivo bioequivalent study?
Yeah, thank you for that question. I would say the thought pattern over here is that we are trying to distinguish a PK endpoint study from say a clinical endpoint study or a PD study. So even a clinical endpoint study would be a bioequivalent study, and so it just helps to clarify and put very clearly that we are talking about the PK endpoint study. Thank you.
Excellent. Thank you for that response. My next question is for MJ. The question is, going forward, can we expect that a single BE study using a comparator, either from EU or USA, will be sufficient for global registration? No need to separate for BE studies for EU or US market?
Sure, thank you. Given that I'm from FDA, I'm not going to say what EMA may or may not do. But as you heard from our presentations, the purpose of the ICH M13A was so that we can harmonize the recommendations from different regulatory agencies. So we hope to strive for that, and therefore the recommendations when it comes to one or two studies for for these immediate release products, we hope that when there is one study recommendation, then there should be some harmonized approach with that. So we would like to believe that we are harmonized, and therefore the generic drug developers should be able to utilize our latest recommendations reflected in the October posting of 2024. And with that, I did briefly mention about how there are some differences of BE guidance recommendations, especially the patient-based versus healthy subject-based studies. We actually have a poster presentation which was presented this year in one of those societies, and we actually looked at all those recommendations of drug products where the EMA already has BE guidance recommendations, and we compared that with FDA's, and we recognize that although there is some difference, the percentage was relatively low, of about 5 or 6% of the PSGs that we looked at. And then we are looking at different safety aspects as well as whatever we found from the new drug applications and the literature findings and all. But moving forward in this aspect as well, in terms of patient versus healthy subject recommendations, we hope to harmonize in this area by further communicating with our other regulatory agencies. So we are going to strive to harmonize as much as possible. Thank you.
Excellent. Thank you for that. And that does look like this is all the questions that we have time for today. I'd like to say thank you to our presenters and panelists for all of the excellent information. I will now turn over the microphone to Dr. Robert Lionberger to provide a few closing remarks for today's webinar.
All right. Thank you very much. Really like to thank everyone who's attending and listening to this, and thank all of our presenters for taking the time to do this. I think one thing I don't want our audience here to miss is how significant a shift this is, not just in the global harmonization, but for FDA specifically. Again, as you noted, 800 PSGs changed recommendations here. I did a brief look and I looked at, the applications that we approved last year, right? And said, of those applications that FDA approved last year, if we had the M13 recommendations in place, how many fewer studies would we have to do? And it turned out to be, around 200 studies that were excess. So this is a huge impact on the studies that the generic industry has to do. And hopefully the generic industry will take this and the resources they save here, they'll move them into developing, more generics for other products, and growing that opportunity, competing for different products, since the costs for entering the market are less. Hopefully this will have a big shift. But again, it's part of our general goal as a science-based agency to make our recommendations and our scientific approaches to bioequivalence as efficient as possible. And I really want to commend the people. So you heard from the three presenters, so Lei, Nilofer, and MJ. Those were our three technical representatives to the ICH M13 process. And they really went through years and years of work on this, spending time in addition to their normal jobs as senior positions in OGD to work on this, to have, not just the meetings in person, but many, many early morning meetings with the global harmonization group to build the sense of consensus, to build the scientific foundations around that. And, and also, internally within FDA of socializing this and preparing this. And I think, one significant aspect of it is, oftentimes at regulatory agencies, it's easy just to say, oh, let's be careful and add more studies. And add more studies and add more studies, and our requirements grow and grow, and that's sort of some iron law of government and bureaucracy. Here, really, I think how significant it is, really a shift to a much more efficient approach. That takes real, courage, it takes savvy internally, it takes building your decisions on strong scientific foundations. And so I really want to point to those three members of the team who really led that effort globally and you know, on things visible externally, but also things internally in FDA. And I think that's a fantastic achievement. If you think of the savings for the generic industry, that can be hundreds of millions of dollars, not just this year, but every year going forward, in terms of, you know, making the system and the generic drug program better and leading to, you know, and helping develop and having a sustainable generic industry that provides products to the American public. And, you know, I also want to recognize that not just the people who developed the M13 part, but this change in 800 product-specific guidances, that's a huge amount of work. The team inside FDA who went through that, you know, we really looked, as Lei and MJ said, looked at all 1,000 immediate release product-specific guidances and used whatever resources we had to develop the information, looked at literature, looked at FDA data. What BCS class is this? What's the solubility of this drug? Is it high solubility? Is it low solubility? Oh, if it's low solubility, is it pH dependent? You know, is it solubility, is it a weak acid, a weak base, or is it always low solubility? We looked at what type of formulations were in the product. We looked at the size of the food effect for products that didn't have food effects in their reference product label. We looked at ANDA submissions to see if we had estimates of the food effect. Massive amount of data collection and discussion around that went into that. And, you know, there's a whole bunch of people to do that. One of the people you heard from today was Chi, our IR team lead. You know, so, you know, she was new to this job, and she really received, as one of the first assignments in that new role as a team lead, you know, now we're going to change 800 PSGs, and you know, really appreciate her effort and her recognition and her expertise and deep scientific expertise and understanding of the formulation design factors and risk factors in this. She's really an essential person to that process. And so really, just in closing here, really appreciate all of the effort, but also in this webinar, you really got to hear from our key experts in ORS who worked on this, but you also heard, as we finished the M13 and we finished the PSGs, you also heard from our colleagues in the Office of Bioequivalence. Because, you know, as I mentioned, you know, the generic industry, you could send in some crazy formulation, and our reviewers, bioequivalence reviewers, have to be looking for that. So they have to also be part of this implementation, saying like, look, you did something that's outside of the range of normal formulations. It doesn't meet the expectations that were in M13. So there's a change also to our review processes. You heard from Partha and Nilofer about, you know, what applicants should do. You know, should I submit? I'm in the middle of this transition. What types of studies should I submit? So again, across the whole Office of Generic Drugs, people working together and building out this system to make this much more efficient. And really, I'm very grateful to be part of this and to be able to represent this. And so with that, thank you all for for participating in our webinar. We'll also look at all the questions we didn't answer and try to make sure that in other communications we address them. And so with that, I want to turn it back to our SBIA colleagues for their final administrative closeout. So thank you all very much for being here.